We've been trying to understand how interactions between molecules might produce polar growth in root hairs. And most of what I'm going to present to you today will be quite a speculative theory approach that makes some predictions, but I'm going to show you some experimental evidence. So it's going to be similar in spirit to Elliot's talk at the beginning of the day, in that we have a model. At the moment, we don't know how close to the truth, whatever that means it is. It could be completely wrong, but it seems to capture the essence of the behavior of the system. So this is our system. It's the Arabidopsis root epidermis, and it's got these root hairs on it. And what we have here is a tissue that's polar. And it's polar because the root hairs are all produced at one end of each cell. So if we zoom in and look at one little root hair cell, each cell makes a root hair towards one end. This is the one single cell here, and here is the end of the cell nearest the root tip, and it's making a hair. And this happens over and over again, which produces evenly spaced hairs. This is an auxin-controlled system. Auxin seems to regulate the polarity of these cells, so I'm going to outline the evidence that, very briefly summarize the evidence that leads us to believe this. So, yep. Here is the first bit of evidence, which was from Ottilie Leiser's lab, where she found that the AuxIAA mutant AXR3 is bald. It has no hairs at all. So without an auxin signal and the degradation of AuxIAAs, we don't see root hair growth. And as Yuji's just brilliantly introduced, it's a good thing we swapped over, because I've been able to skip a load of stuff I would have said. We know that we have these pin proteins controlling auxin flow, and they're giving the tissue a polarity, and I'm going to summarize some of that in a minute. And then we know from published work that auxin is detected in the nucleus by the degradation of these AuxIAAs, such as AXR3, which I just showed you, and this can liberate auxin response factor transcription factors, and they regulate auxin response genes, and we get responses. So we know that auxin matters for root hair production, and we wanted to understand this polar system with these root hairs growing at one end of the cell only, and to start with, we needed to know what this auxin flow pattern was in our tissue. So I'm going to tell you about that first. So here is a diagram which Anharad Jones drew of the root epidermis, the root surface, and we have two cell types. We have these purple cells, coloured purple, that don't make hairs, and then we have rows of cells that do, with these non-hair cells in between. If we cut away and look at where the auxin flow is, this is summarising lots of data from many labs and many papers. Auxin is being pumped down mostly from the shoot. Some is being locally synthesized as well. Then it's being pumped up because of the localization of pins up the outside. Malcolm showed some data on this earlier as well. And then it gets refluxed back round. So a little bit of it escapes up through these developing hair cells, and most of it is recycled. And as this meristome grows away down, the cells are left behind, and the pins are reorganized such that we maintain this pattern in this growing root tip. And as the cells get older, as they're left behind, they're experiencing a changing auxin environment. If we dim the lights, please. So we needed to know in detail where the auxin transporters were in our system, and that had not been published before in the detail I'm going to show you now. So we looked carefully at where auxin influx carriers and auxin efflux carriers were, and we found that the pin 2 efflux carrier is always at the top end of the cell. So if we zoom in and look closely, this is the top end of the cell furthest away from the root tip, pumping the auxin up. That had been published before, but what hadn't been seen before was that AUX1, the influx carrier, is actually localized all around the non-hair cells, but is not in the hair cells. So this was a big surprise, because I showed you earlier you need an auxin response to get hairs at all, so you'd expect the hair cells to want to see an auxin signal, and yet they're not influxing auxin. They're allowing the cells next door to do that. And to understand what was going on here, we needed to go beyond just intuitively interpreting these images, and we needed to build a simulation, because we can't directly observe auxin, yet we don't yet have any of Wolf's fantastic sensors that might let us see where auxin is in the wall and inside the cells. Instead, we've had to use computer simulations to predict where the auxin will be. So we've done that with Eric Kramer by building a three-dimensional model of the root. So we built a three-dimensional model with the AUX1 around the outsides of all these cells. It goes down, not just the epidermis, it goes through the cortex, the endodermis underneath. We've got the pins at the top end, furthest away from the root tip. And that gives us a readout of auxin concentration through the developing cells. And on this graph here, we have a plot of the output of the model. This particular plot is looking through the middles of the cells. It's looking through, so if we're going along, here's a diagram down here. You can see this is a little cell here, and then there's a big sharp peak of auxin, which is in the wall between two cells. Then we get the next cell, the wall between with it and the next one, and then the next cell. Okay, so the first thing that we got out of this output was that the non-hair cells are stably maintaining a very high auxin level. These are the ones expressing the AUX1 influx carrier. The hair cells are stably maintaining a much lower auxin level. And I'm not going to go through, because of time, the details, but if you remove AUX1 from the model, what happens is that all the auxin gets taken back down into the root tip. The cells lose their auxin signal early. This was published in Nature Cell Biology a couple of years ago. And what you get is short root hairs because they run out of auxin prematurely. So what's happening here is that these non-hair cells are acting as sort of leaky straws. The pin means they suck auxin up the root towards the shoot, and the AUX1 allows them to hold it, but some escapes. And in the absence of AUX1, enough of that can cross into the root hairs by diffusion, into the root hair cells by diffusion, to sustain root hair growth. So it's a drip feed system where the root hairs are irrigated by the non-hair cells next door. The second thing we got out of this, which we didn't comment on in our paper, was that there seemed to be gradients of auxin across each cell. So the internal auxin concentration is much higher. This is a log scale. It's much higher at the end of the cell nearest the root tip than at the end nearest the shoot tip. And we were interested in this because for a really long time we'd wondered about this polarity question, why the root hairs are always at one end of the cell and not the other. So that's what I introduced earlier on. So we've got this hair towards this end of the cell. So what we discovered was there's an auxin gradient across this cell. High auxin here, low auxin here. To think about that, we needed to zoom in to one cell at a time and think about the molecules and their interactions at the start of growth. And we also had a lot of wonderful historical data from Marcus Grieber's lab and from our own lab, where we'd manipulated molecules that affected this process. So it was already in the literature from Marcus's lab that the AUX1 mutant could make multiple hairs. This is one cell. These little arrowheads tell you where the ends of the cell are. It's making more than one hair. This is data from our lab showing that if we overexpress ROP-small GTPases, we can get multiple hairs. And we know about hundreds of molecules in these cells, but we decided to focus on the ROP system because we knew it's sufficient to drive growth. If one can localize the ROPs, one can localize growth. We knew that because we'd done some work adding extra ROP, overexpressing the ROP protein in root hairs, and we can get much longer root hairs. This is root hair length. This is data from nearly 10 years ago. So we can keep the root hairs growing just by having more of this ROP. Not only that, we can get extra hairs. So a single cell will try up to 7 times to make hairs if it has enough of this ROP. So we were trying to work out if we could put together this Auxin gradient with what we knew about ROP molecules and understand how the two might affect each other. What we know about ROPs from Yulowski's lab, is that when they are active, the class of ROPs that we're dealing with in the root hair system, they are S-acylated. So they have an extra lipid attachment, which is widely believed to affect their movement. And in the light of John's talk, maybe we need to think about whether the wall whether there's a knock-on effect to interactions with the wall here. But certainly it's been shown experimentally by the Yulowski lab that activated ROPs are less mobile than inactive ROPs. And that this acylation is important for that. We also knew that patches of ROP appear, very discrete, predictable patches before growth starts. So we wanted to know if with this information about ROPs and their movement, depending whether they're active or inactive, with some information we could get from the literature on how active ROP might influence inactive ROP and its activation, and with our knowledge of the Auxin gradient, we could get any insight into this polarity. A very important piece of evidence came from Marcus Grieber's lab, beautiful experiment where they found that if they disrupt auxin synthesis and transport enough, and then apply auxin at the wrong end of the cell nearest the shoot, they can move the hair. So this really clinched the idea that this Auxin gradient that we'd seen really was controlling where ROP patches formed. The ROP patch was forming first, and then the hairs were growing. So this gave us more confidence that this was worth thinking about. So we built a theoretical model. We built a theoretical model, and the main motive for doing that was initially that my mathematical collaborator, Robert Payne, wanted to prove to me that the ROP system plus the Auxin gradient could not produce patches such as we saw. So we built the model for fun to find out how far away it was from working and what else we would need. We had Auxin gradients along the cell. We had plausible estimates from the literature from comparable proteins of diffusion rates for inactive ROP and active ROP, and then we tested. I'm not going to show equations because it's late in the afternoon and I hope that it will speed us along. But we simply summarized this diagram in partial differential equations. And this is based on existing knowledge of ROPs. So ROP and its relatives, certainly its relatives in other species, the small GTPases, if they're active, they can recruit inactive ROP that's nearby. And there is an auto-activation effect. You have regulators which encourage the inactivation of ROP and the activation of ROP. We have in our model the rates of delivery of active and inactive ROP locally to a part of the cell. We have removal of ROP, and we have the fact that this inactive form could move around much more quickly than the active form. And first, we tested whether or not auxin could be acting on this autoactivation step, because in yeast, when they bud, this is the step where regulators are acting to produce patches of ROP-related small GTPases in the yeast cell. So I'm going to show you an agent-based model first. So here we've just implemented this model with little agents, little blue dots moving around quickly to represent the inactive ROP, little red dots which are activated ROP. And we're just going to implement those rules. So the green is the Auxin gradient, higher Auxin at this on the left, lower Auxin on the right in a gradient. And we simply tell these little agents that if you're blue and you're in higher Auxin, you're more likely to go red. And if you're near a red ROP, you're more likely to go red. What that shows is that even with this ridiculously crude model, an Auxin gradient, such as we see, combined with what we know about ROP behavior, can produce a patch. Every modeler knows that if you choose the right parameters, you can get your model to produce all kinds of nice behaviors, but it doesn't tell you that it's really the essence of the model that you've built. And so we actually did some analysis, and we looked for the boundaries between different states in these equations. So we solved them and look for boundaries and look for the behavior of the equations across lots of different parameters, because we've just estimated parameters here. And what we found was that we have a range of auxin concentrations, a range of cell lengths under which we would predict the root hair is going to form right at the end of the cell. Another range where we'll get wild-type phenotypes, a range where we'll get 2 hairs, and we can map out the range of things that we might see. We also found that the Auxin gradient can be incredibly weak, and we will still reproducibly get these patches in the model. We only need a 10% difference in Auxin locally to produce a patch. So it's a really robust system. And also, I'm not going to show it today, but there is hysteresis in the system, which means that once a patch forms, if the auxin then disappears, the patch doesn't disappear. It persists. So it's robust in many ways. Okay, so far so good. We're thinking that auxin might act at this stage, but because we wanted to do this thoroughly, we tested auxin on all the other steps. And the only other place where auxin could be acting in the model and produce the results that we're seeing and the types of phenotypes we see is at the localized delivery of inactive ROP. None of the other places give the range of phenotypes that we see. So we get a similar plot for this version of the model, where we have a range of auxin concentrations and a range of cell lengths where we can find either no hair or a hair right at the very end of the cell, or the wild-type phenotype, or 2 hairs. So we can say from this that it seems that we would predict at the moment that auxin is acting either on this autoactivation step or on the local delivery of inactive ROP at a particular site in the cell. Okay, so we have a very simple theoretical model. It's well behaved. So what? Does it make any nice predictions? It predicts that we have a range of phenotypes and that we can move along these by manipulating things like cell length, Auxin gradient, ROP level, activity of ROP regulators. Here are some examples. So it predicts that cell length will affect hair position and that we should see a relationship like this between hair position and cell length. And there's lots of data on this in the literature already. So we went to the historical data, mostly from Marcus Grieber's lab, and we analysed it for this relationship. And we get really nice probabilities for the data lining up with this kind of relationship. We have 2 different groups of data here. Nearly all the mutants and transgenic lines made are lining up, where these things have been measured, are lining up along a line here. But this group here stands out, and these are all GNOM, or combinations of GNOM mutants. So this suggests that in a GNOM background, sensitivity to cell length is an order of magnitude different than in other backgrounds. Another prediction, plants with more double hairs will also have more hairs at apical positions. So this is where the hair shifted along the cell. Again, this is nicely satisfied by the historical data so far. Another prediction, this one's a bit more complicated to test. High auxin will produce apical shifts and double hairs. Here we had to go to a completely unrelated mutant that had never been tested for pattern before. We had to find a mutant where we expected high auxin in early young hair cells. And the mutant we chose is the Werewolf MIB double mutant. This is mutant in 2 transcription factors that affect root hair patterning such that there are no non-hair cells, there are only hair cells. This means that these epidermal cells don't express any AUX1, and all the auxin gets trapped near the root tip. The auxin gets trapped near the root tip. It is quickly recycled as we're moving back from the meristem. And so we expect in this mutant high auxin in the young cells. So we'd expect this mutant, which is a root hair patterning mutant, to actually have more apically shifted hairs and double hairs. So we looked at that, and we have more apically shifted and 40 times more double hairs. And the final prediction I'm going to summarize today is this one, that a patch of ROP should start at the basal end of the cell and then it should move. And Harry Jones and Harriet Jones collected some data on this, and the patch of ROP does indeed start at the very basal end of the cell. This line is indicating that wall, and it does drift, as predicted by the model, away from the end. So we are able with this model to reproduce a whole suite of mutant phenotypes, loads of them, historical data from the literature, new data that we've collected ourselves. So perhaps we have the beginnings, the essence of a plausible explanation for how high auxin might act locally and influence the accumulation of a tethered patch of ROP, which, because it can recruit more ROP, or in the alternate version of the model, because local ROP delivery is affected, produces a patch that produces polar growth. The mystery in all this work, though, is that this would all require a new auxin receptor, an intracellular auxin receptor that could regulate ROP activity according to the intracellular auxin concentration near the plasma membrane, and no such receptor currently exists. We have considered the possibility that ABP, which is believed to have sense auxin extracellularly, could be influencing this system. With the current models of Auxin flow in the wall, that wouldn't fit, but we will continue exploring these possibilities as we can. So, I need to thank the people who did the work, who are listed here: Harry Jones, Victor Brenner is a mathematician who's working on the maths of our model, and Robert Payne, who was very important in all of this work, Eric Kramer, who produced the simulation of Auxin flow, and the other authors here, and also Yeji, who's in the audience because Harry went and spent some time in his lab looking at ABP amongst other things. And that's it.